Right. Good evening.
Good evening, sir.
Right. Great. Okay. I think the other student, she won't join today. That's fine. We can start. Right. So today we have to start the new topic that is on photosynthesis. Right. So first of all, let's understand what is the definition and what is the idea behind photosynthesis. Right. Photosynthesis. Photo means it refers to light. Synthesis means producing something. So mainly plants, since they are autotrophs, they only produce food. They produce food, but they do not require food. They just produce food. So they don't depend on other organisms as well. So the word photosynthesis, you can get the idea. Photo means light. So using light as a main source, the sunlight, plants will produce food. Right. Okay, the student is there. Okay. Right. So photosynthesis. Photo means it's the light. Synthesis means production of food. Right. Today we are going to study the basic raw materials that are essential for photosynthesis and the practicals related to photosynthesis. Okay. Now first let's go through. Yes, you need to get the idea about photosynthesis, especially um, the autotrophic and heterotrophic. Autotrophic means where the plants produce their food within themselves, which is known as autotrophic nutrition. Only plants are autotrophic, but when it comes to animals, they are heterotrophic because they depend on other animals and or other plants, right? So you can clearly see that it's mentioned as heterotrophic, right? Okay, understood that part? Hello? All right, I'll look at the you didn't understand. Right, okay. Right, let's then repeat it again. Fine. Right. So first then let's start from the beginning. Okay. Photosynthesis. Hope you can hear me well. If there's any audibility issues, let me know. Right. Photosynthesis. Right. I'll use the board to explain the photosynthesis. Hope you all know what is a photosynthesis, the process of photosynthesis. Photosynthesis. Okay. Right. Photosynthesis is only involved in plants. Right? So it's only involved by the plants because plants only can produce their food. Photo means they refer to light. Photo. Oh, give me 1 second. Because there are some students are waiting for the trial. That's the thing. Okay. All right. So photosynthesis, right? Then it produces light, and then synthesis means producing something. So during the process of photosynthesis, you have to clearly understand that with the help of light, with the help of sunlight, they produce food. Okay, right. Now, did you understand that, Shima?
Yes, sir.
Yes, you understood that, right? Then the other part we were discussing is about The modes of nutrition. The modes of nutrition. Right. There are two forms of nutrition. There are two modes actually. You can classify as the number one. It is autotrophic. Autotrophic. Actually, the word auto means it automatically makes its food. It produces its own food with the help of sunlight. So plants, every plants are autotrophic except few plants. They are heterotrophic. They cannot produce their food. So they sometimes depend on other types of plants or other organisms. That's an exceptional case, but you have to mainly understand plants are autotrophic, so therefore they can produce food. Right. Then number two, the modes of nutrition, it is heterotrophic, heterotrophic, which is known as they cannot produce their food, therefore they depend on other organisms, depend on other plants or animals. So that's simply we say it as organisms. So did you all understand about the modes of nutrition, the autotrophic and heterotrophic? Is that clear? You can send the chat messages as well if you cannot speak through the microphone. If it's not clear, you can feel free to ask to explain it again. It's clear, right? What about the other student, Chenumi? Chenumi, actually, we were discussing about the modes of nutrition, right? So based on the modes in sense, when the plants, they produce their own food, so they can produce their own food using the sunlight. So therefore, they are known as autotrophic. But when it comes to heterotrophic, what happens? They cannot produce their food. So therefore, they are known as heterotrophic. They depend on other organisms. Is that clear? I think it's clear for Shima. What about Chenimi? You can send a chat message. If it's not clear, it's clear, right? Good. Now, actually, our focus is not about to study about these modes of nutrition. That's the basic idea behind photosynthesis, right? First, let's study what are the raw materials or the factors that is necessary for the photosynthesis to occur, right? Now, simply they have given a picture. So this picture illustrates that these are the different factors necessary. So by the word photosynthesis, you can understand they need light as a source of energy, especially the sunlight. And then plants are green in color, so therefore they contain chlorophyll pigment that's green. So because of the chlorophyll pigment, the sunlight gets trapped into the chlorophyll pigment, especially that chlorophyll pigment is present inside the leaves of the plants, right? So then sunlight is trapped into the leaves and where it helps in photosynthesis. So we now discussed number one, sunlight as a source of energy. Number two, chlorophyll. So when it comes to chlorophyll, what happens? Chlorophyll is a green color pigment which traps that sunlight and helps in photosynthesis. Okay. Then the number three, the two main raw materials. Normally for the plant growth, we need water. So water is absorbed by the root hairs. You can see here the root hairs, and the roots are actually present under the ground. So therefore the roots help to absorb the water and the mineral ions which are present under the soil. So they need water, sorry, they need water as well. And then they need a gas, which is an important gas. It is known as carbon dioxide. Normally, you have to understand the carbon dioxide gas is released, sorry, it's absorbed through the stomata. Do you all know what is known as stomata? The stomata, plant part, actually this is a guard cell, and the pore which is present in a leaf, the stomata. Shima, do you know what's a stomata? Shall I show you a picture on stomata? What about Chenumi? You want to see a picture, right? Okay. Then let's look at a picture. Actually, it's the microscopic part. It's microscopic in sense you cannot observe without the microscope. Stomata of a leaf. Let's look at a picture to understand the morphology of it, the external features. Right. This is how the stomata looks like, the microscopic structures, but this is how it works. When the carbon dioxide or water enters into it, actually the stomata will get closed. The mechanism, we say, the method of functioning, that's known as mechanism, right? Okay. So this is too much for you, but it's like when the stoma becomes open, then the carbon dioxide will easily enter towards it. And when the stoma is closed, actually carbon dioxide cannot enter. This is how even the gaseous exchange happens, especially the plants, they do respiration. So, and photosynthesis as well. Photosynthesis, they need carbon dioxide, but for respiration, they need oxygen. So all of these gases get diffused, move through the pores called stomata. You can't see it? Oh, okay, sorry. Right, this is what I referred to. All right, sorry. Yeah, this is the picture which I referred to. Okay, so this is the pores which I mentioned, the stomata. Now in this condition, the stoma is open. So therefore, the gaseous carbon dioxide or the other gases, they can diffuse in, right? But in this case, the stomata is closed. So therefore, the gases cannot diffuse in, right? Understood about that? That is an important part, knowing the stomata. Is that clear? Okay, fine. Then I'll move to the next part then. Right. So what were the raw materials we discussed? It's clear, Chenmi. Okay, great. Right. So yeah, what about Shima? Is that clear? The stomata part? Uh, were you able to look at the picture? Yes, it's clear. Right. Okay. What were the four main raw materials we studied? Number one, chlorophyll. Number two, sunlight as a source of energy. Number three, water. Then number four, carbon dioxide, which enters through the stomata. Okay. And when you look at the definition of photosynthesis, this is a very important definition, utilizing the light energy. So by using the raw materials, you can construct your own definition. Let's look at it. It's so simple, utilizing the light energy of the cells containing chlorophyll. I said, so when the light energy is only trapped with the help of chlorophyll, which is in green plants, it's in green plants, which synthesizes the food. And remember, students, the synthesizing word means it's producing the food, right? Do anyone know what is the form of food that's produced in plants or during photosynthesis? You all can send through the chat. The form. Otherwise, you can raise your hand to send the message or the answer. What is the form? No one? Okay, right, that's fine. So the form of sugar that is made during photosynthesis is known as glucose. Now you have to remember that name. It is glucose, right? Glucose is the form which is made during photosynthesis, right? Is that clear? Now I started from the basics with the word photosynthesis. Then we studied about the factors, the raw materials or the factors that are necessary for photosynthesis. They were solar energy, that's the sunlight, then carbon dioxide, water, and chlorophyll, which chlorophyll is a green color pigment. Remember that. That is the reason where the leaves are green in color, because it contains which part, dear? Is it the glucose part or the photosynthesis? This part, the definition. Chenemy, is it the definition? Hope I think so. It's the definition. The glucose part. All right. Okay. Yeah. So now we have studied what is the definition of the photosynthesis. It says clearly utilizing the light energy, the cells containing chlorophyll. So the leaves, actually the cells means the leaves, the plant cells, they contain chlorophyll. So chlorophyll is green in color. The green color pigment absorbs the sunlight, absorbs the light energy, and synthesize food using carbon dioxide and water. Now I'm going to highlight the synthesizing food. Now, first my question was about what is the special form of food they produce? That is known as glucose. Glucose, I think you all have learned in your grade 10 syllabus, where the glucose, she left. Okay. Right? So the glucose, it is a simple carbohydrate or a simple sugar. Okay. Right. Understood, Shima, about that?
Yes, sir.
Yes, you understood. Right. She asked the question and she left. I don't know why. Maybe due to a connection issue. Okay, that's fine. Okay. Right. Now, today we have to study, as I mentioned, we can look at here the factors that affect the photosynthesis. As I mentioned, what were the factors? Chlorophyll, light energy, water, and carbon dioxide, right? So all the four factors: chlorophyll, light energy, water, and carbon dioxide, right? Okay. Now let's move to the other part. Actually, today our day is to study about different investigations, different practicals to test whether are these requirements, are these factors necessary for the photosynthesis. The four factors. So there will be four different practicals, right? Okay. And then there will be the fifth practical where we will be understanding, especially when you look at the equation of photosynthesis, this is an important equation, right? This equation clearly states that where carbon dioxide is essential for the photosynthesis and it combines with water, which forms light energy and chlorophyll, right? Light energy and chlorophyll, sunlight and chlorophyll, right? And then what is the food that is produced? It is the glucose, right? So glucose. Then when it comes to oxygen, it is a byproduct, we say. It's a byproduct. It's a gas which is released after photosynthesis. Okay, right. So even there's a practical to investigate whether oxygen is released during photosynthesis. So there are 5 investigations we have to learn. Okay, understood that part?
Yes, sir.
Yes, that's clear now. Right. Okay, then we will start one by one. The first yeah, before that, we have to just understand about the products of photosynthesis, right? Are you able to read this section?
Yes, sir.
Okay, dear, you can read it then.
Products of photosynthesis. The glucose C6H12O6 produced during photosynthesis will be temporarily stored in leaves as starch. Later, part of that starch is converted into sucrose and transported into the other tissues. We phloem. The sucrose that is transported to the storing organs are again stored as starch.
Right. What are the different examples?
Vegetables,
Fruits.
yams, leaves, roots.
Right. Fruits, vegetables, yams, leaves, and roots. Right. First, you have to understand, as I mentioned, the glucose is produced during photosynthesis and it is temporarily stored as a starch and it is converted into sucrose. This is the form. Actually, I'll use the board. Glucose is produced. Then it is stored in the form of starch. And it is transported throughout the plant as sucrose. So this is an important thing. Produced form. This is the produced form. And then the starch is the stored form. And then the sucrose is the distributing form, or we say in other terms. Oh, she's joining again. Okay, so when the sucrose is actually, that's how it's distributed throughout the whole plant. Okay, right. Did you understand that part? Okay. Right.
Yes, sir.
Okay, great. Jennifer, we were discussing about, I can see that you have a connection issue. That is fine. You can try to fix it by next week. Right. So glucose is the one which is produced during photosynthesis. That is the pure form where it's produced during photosynthesis. Right. And then where after it's produced, it is being stored in the plants. So what is the form or what is the name given or the chemical, in which chemical form is it stored? That is known as starch. Okay, right. And the stored form is starch. Then after it is stored, actually it should be distributed throughout the plant, transported throughout the plant, we say. So translocation in the first topic, Shima, we studied, isn't it? The phloem involves in translocation. The food products are being transported throughout the plants. So that is in the form of sucrose. So these three types of chemicals are very important. Glucose, starch, sucrose. Okay, right. Hope that's clear. Right, now let's move to the other part. Right, and Shanami, we were discussing about the equation for photosynthesis. This is a very important equation. Right. So with this equation, you can get the idea what are the necessary factors or the raw materials we need for photosynthesis, especially carbon dioxide and water we need for photosynthesis. And when it comes to the other sources, especially chlorophyll, pigment, which is necessary for photosynthesis, light energy as a source of energy, sunlight, and then the products form. Normally, you might have studied in chemistry section, the chemical equation part where the reactants are reacted onto the left hand side and the products are written on the right hand side. So these are the products formed during photosynthesis. The main product is the glucose and the byproduct we say that is the oxygen gas. Okay, but our target, our focus today is to study, to investigate and study the practicals related to test each of these factors. Now let's say Now, according to the equation, according to theory, they have clearly mentioned that they need carbon dioxide, they need water. Now, how can we prove it? That's the practical investigation we are going to do. Okay? Right. 1 second, there's another student. All right. Now let's start our practicals. Okay, students, is there any doubts? Because you have to clarify all your doubts before we go to the practical part. Is that okay with the definition of photosynthesis, the glucose, starch, sucrose, and then the equation? Is there any doubts? Please feel free to say. You all can just send some quick chat messages. Got it. Great. Okay. Yeah. Okay, that's perfect. Yeah. Right. Then fine. We'll start our practicals. So I mentioned that clearly there are 5 practicals. So you can see there are 4 raw materials that are essential for the photosynthesis. You can look at here. Number 1, chlorophyll, light energy, water, carbon dioxide. So we are going to do all these investigations. So 4 practicals will be done to test whether these raw materials are important or not. And then the other thing is extra one, I said the byproduct of photosynthesis. It is the oxygen. We are going to test whether during photosynthesis is the oxygen released or not. Right. Let's start our practicals. And I'll try to guide you all with some videos of some practicals. Right. Now the first one, we are going to identify starch produced during photosynthesis. This is actually known as starch test. Right. Right. The materials required, you need a beaker, a test tube. So you all are knowing. Hope you'll know about a beaker and a test tube and a tripod. Tripod is kind of like a stand where you place the Bunsen burner. This is the Bunsen burner used to heat the solution, heat the mixture. Some amount of water, alcohol, and plant leaf. Okay. So we are going to indicate. Now we said that plants store the form in the form of sucrose. They store in the form of sucrose. So therefore, you have to understand here the starch test. Sorry, store in the form of starch, right? So the starch test is done. Is it true or not? The validation is done using this investigation, right? What's the simple methods we do? The number 1, we pluck a leaf from a plant which was in sunlight and boil it in water. So a leaf, a normal leaf you can pluck, boil it in water. Okay. Then place the leaf in the boiling tube with alcohol and boil the tube in water bath for few minutes, right? So then you have to place the leaf in the boiling tube with some amount of alcohol. What is the reason we are going to put alcohol and boil it? Because our objective is to remove that chlorophyll. To remove chlorophyll. The chlorophyll pigment should be removed. We are going to test only the starch in it. And then finally you wash the leaf. Then you have to be very careful because there can't be any remaining alcohol in the leaf. And you boil the tube. Therefore, after you boil it, then every remaining portions of the alcohol will be not there. Then what you do, you start to add some few drops of iodine solution. Hope you all know about iodine solution. It's a kind of a brown color solution. Then when you add it, what happens? Suppose if the starch is present, those are the food tests you need to know. Grade 10 syllabus, the first topic, Burette, sorry, Burette test, Benedict test. So similarly, starch test, where iodine is added to a drop, some few drops of iodine is added. So therefore, there will be a color change that occur. The color change will cause to say that whether is there starch present or absence, confirms the presence of starch, right? Confirming the presence of starch. So normally when it turns to blue-black in color, that shows there is starch in it. But if there's no color change, a blue-black, or they say blue or dark purple as well, the leaf turns. This is the main observation. If the leaf turns to blue or dark purple, then the iodine is added. It's concluded that the starch is present. Yes, we have to boil the leaf. That is because to dissolve the cells and all the chlorophyll. That's why we are going to boil the leaf, right? Okay, that's good. Chenumi, do you have any doubts regarding this first practical? Anything to repeat? Okay. No, it's clear, right? Great. So why do we do the iodine test? The main thing is to test whether the presence of starch in it or not, right? Now let's go and remember, students, you all have to know that when it comes to chlorophyll, actually, why do we add alcohol and boil it? To dissolve the chlorophyll, right? So because our objective is not to, I want to explain it again. All right, okay. Okay, I'll brief it, the experiment again, right? So this is the activity, right? Why do we do this activity? In order to test the presence of starch in a leaf. Okay, please mute your microphones. Right. So how do we test the presence of the starch? Right. First, what you do, simply take a leaf, which is like exposed to sunlight, and boil it in water. So when you boil it, what happens? All the cells will be dissolved. We need only the starch. So therefore, the first thing you boil it. Then second step, you have to boil it with adding alcohol to it. So why do we add alcohol? Therefore, the chlorophyll, the green pigment layer, will be fully removed. Okay, right. Then third step, what you do, you wash the leaf and put few drops of iodine. You have to wash it clearly, and remember there can't be any remaining of the alcohol in the leaf because it might affect your results. The color change might not occur, even though if there's starch present in it. So therefore, you have to put, then after washing it carefully, then you have to add few drops of iodine. Iodine is a solution of kind of a brown color solution where you can observe the change. If there is a color change occur, you can clearly see that it should be turning to a blue or dark purple once the iodine is added. But if there's no color change, it indicates the absence of starch. Right. Is that clear for everyone? Shima, is it clear now? Got it, the practical, because these are important practicals. It's clear. Great. Okay. If there's any doubts with the other students, you all can feel free to send a chat message. Right. All right. That's the first practical. Right. I'll try to support you all with some videos. Yes. The one who raised the hand. Is there any doubts? You all can send it to the chat. Okay, right. Testing of factors required for photosynthesis, right? Now let's look at the other factor. We are going to check whether sunlight, is it essential or not, using the second activity, right? Now what we do is simply, you take a potted plant. This is an example of a potted plant, right? And keep it in dark for 48 hours. So carefully listen, this is very important. You have to keep it for about 48 hours, right? So that's around like 2 days. But keep it in dark, like in a dark cupboard where there can't be sunlight or any light entering it, right? So then what you do, the materials, the extra materials you need to do the starch test. Yeah, normally you have to carry out the starch test as well, right? And then some polythene strips and a black color and colorless polythene strips are essential, right? These are the things needed. Right, now let's look at the method. Just select two leaves of your potted plants. You can name it as A and B, right? Of the plant kept in dark for 48 hours. Now let's say you can select any of the leaves. Now in this case, they have selected these two leaves, A and B, right? And cover the part of leaf A with black polythene and part of leaf B with colorless polythene, right? So the one part you have to cover with a black color polythene or kind of a black color paper. And then the second part, maybe the B part, you can color with a colorless polythene, right? Then what you do, expose it to the sunlight for about 3 to 5 hours. So when you logically think what will happen, for example, when it's a black color, definitely the sunlight, now you have to think about whether the sunlight can be absorbed or not. Normally when it's black color, the light cannot be absorbed that much, not at all actually. But when it's a polythene, the light can be absorbed. Let's see what are the investigation conclusions, right? Now you can clearly see that there's no color change, and we do the starch test. Now students, you all have to remember, now normally we don't repeat the thing starch test again and again. Simply this is the process of a starch test, right? Then after you do this, you have to carry out the starch test separately for the both leaves A and B, right? Then after you carry out the starch test, what are the observations you will get? So why do we do the starch test again? To indicate the presence of starch or not, right? So you have to add the iodine and everything, the same procedure, right? Then you can look at the structure of the leaf A. The one leaf A is actually, students, it's a black color. So as I mentioned, since it's covered with black color polythene, it cannot absorb sunlight. So you can see there's no color change. So there's no photosynthesis at all, right? Then when it comes to the B part, that has converted to a dark purple color, right? So when it's converted to a dark purple, it indicates the presence of starch in it. That is the important thing, right? Is that clear for you all? So this practical gives us the idea and validates that light energy is essential for photosynthesis. Without light, photosynthesis cannot occur. Is that clear? Everyone just send a quick chat messages. Okay, then hope that's clear. One student have sent the message. Okay, what about Chenumi, Asmat? You all can just send a message whether is it clear or not. That would be really helpful for me. Okay, 2 students have sent now. It is clear. Great. Okay, fine. Right. So that's what the main objective of doing this practical. Good. Now, actually, students, these practicals are easy, but in the exam, they don't give easy questions. You have to use the knowledge of this practical and apply for your questions which are given. Normally, they can give different questions. Why they have covered with black polythene? Right. That's because to as a control setup. That means black polythene means it can't absorb any light. So that shows the colorless polythene, it can absorb sunlight. That's the reason, right? And let's go through this part. Actually, the covered area of leaf A has not received the light energy because it's covered with a black polythene. Therefore, photosynthesis cannot occur. Hence, there is no color change with the iodine solution. So for every test, we do the starch because that is a final confirmatory test to check whether photosynthesis occurs or not, or the presence of starch or not, right? Now, students, let's go with the third activity. That is the experiment to show carbon dioxide, which is required for photosynthesis, right? In this practical, actually it looks similar to the previous practical, but the materials and the chemicals we use is a bit different. Now, the first thing what you need to understand, again, uh, you have to use a potted plant and the materials that are required for the starch test. Again, you're going to carry the starch test because there's a confirmatory test and 2 polythene bags are there. Then you have to cover the leaves as such shown in the figure and you have to cover it with some polythene bags. But one of the polythene bag, you have to fill it with water. The other one, you have to fill it with potassium hydroxide solution. Right. Now, I'm going to just a little bit explain about this potassium hydroxide solution. KOH, we say the chemical formula is KOH. What is the purpose of adding KOH into this bag, one of the bag? Let's say in this case, it's bag D or the leaf D. Why? Do anyone have the idea why we add KOH? No one, right? Yes, so the purpose or the objective of adding KOH is because to absorb the carbon dioxide. So it's like a control experiment they do. One side with water, so therefore carbon dioxide is not absorbed, but in the D leaf or the second side of the leaf, the D, as it's mentioned as a D, they absorb every carbon dioxide around it. That is the reason of adding KOH. Normally, we call this KOH as an absorbent of carbon dioxide. Okay. Hope that's clear for you all. Now let's look at the methods. Simply what you do, select 2 similar leaves of the above plant C and D. A similar leaf should be selected. Adding potassium hydroxide and water to the transparent polythene bags. Remember, in this case, you never use a dark polythene bag and a light polythene bag. You have to use only transparent polythene bag because we are not going to test about light here. We are going to test about carbon dioxide. So to get our results well, you have to add KOH. That is the important part. Then after adding KOH, then after some time, you get those leaves, detach those leaves. Detach means remove those leaves. Place it in the sunlight for about 3 to 5 hours. And then you have to detach those leaves and carry out the starch test separately. So the starch test is a confirmatory test where you all can understand the color change. What would be the color change? When it comes to D, there is no color change because you can look at here the D1 is with potassium hydroxide because that shows carbon dioxide is very essential for photosynthesis to occur. So therefore no color change. If there's no photosynthesis, there's no production of starch as well. But in the case of C, there's no issue. There's enough carbon dioxide available. There's water as well. So as a control, they have used water. So photosynthesis occurred. And after you carry out the starch test, it converts into a dark purple or a blue color. So the color of iodine is kind of brown. If it remains brown like this or kind of yellowish color, that means no color change. The same color of the iodine will remain. But if it change into a dark purple or a blue color, it indicates the presence of a starch. And also it shows that the plants need carbon dioxide, which is as an essential factor for photosynthesis. Right, students? Is that clear about the the carbon dioxide investigation? Now we have done 3 practicals. So actually the starch test is a common test. So for every investigation, we carry out the starch test. Number 1, we studied about uh, light intensity. Now number 2, carbon dioxide. Are those both practicals clear for you all? The KOH part is not clear, right? KOH is not that it's not that hard, okay? It's not complex as well. You have to understand that KOH is a kind of a solution. What does it do? The main function of KOH is where it will absorb the carbon dioxide. Why should it absorb the carbon dioxide? Then only you will have a control experiment to compare the results. So when it absorbs all the carbon dioxide, there's no carbon dioxide. So therefore, that's why the reason the leaf D has no color change. Okay. Now is it clear, Shima? What about the other student? Amar? Oh, Asmat. Y'all can send a chat message. It's clear, right? So the importance of KOH is to absorb the carbon dioxide. Right, clear. Okay. Right. If there's any doubts, feel free to ask it. Right. Now, we have studied investigation of light, investigation of carbon dioxide. Then the starch test is the common test. That's to identify the presence of starch in a leaf. Right. Now, the third practical, students, we are going to move on with is that chlorophyll. What is the color of the chlorophyll pigment? It is normally in green in color. So even according to the equation, they have clearly mentioned that, you can look at the equation where they have clearly mentioned these are the factors which is essential for photosynthesis. Carbon dioxide, water, so they need carbon dioxide in water, as well as additional factors, sunlight as a main source of energy, and chlorophyll as a green color pigment. Now, our tests, we have done the investigation on carbon dioxide, light energy. Now we are going to do on chlorophyll. Right. Now let's go through the practical on chlorophyll. The necessity of chlorophyll. This is actually a simple practical. These are the materials you need, especially for investigating the chlorophyll, which is required for photosynthesis. Simply, you need a mosaic plant leaf. Students, mosaic plant leaf means it's kind of like a croton leaf. I will show you some pictures, share in the screen, yeah, of a mosaic plant leaf. And there are some videos also ready for you all to look at about these practicals, right? Actually, the mosaic plant, the name has given because there are some patterns which can be observed in the leaf. It's not working. Okay, I think I'll show it later because it's not web browser is not working. The reason just loading now. All right, now fine, yeah. Mosaic plant leaf. Normally, it's can be observed in a croton mosaic pattern in a leaf. This is a very important thing, students. Yes, this is normally mosaic actually. This is like more artificial. When I show you a croton plant, hope you all know that a croton plant, croton or hibiscus, croton plant leaf. Okay, this is what I mentioned. So you can see here, there are some areas where they lack chlorophyll. You can look at here. When we take an example of leaf, yes. So these areas, some areas are green in color, but some are not in green in color. That's mainly because of they lack chlorophyll, where there's no chlorophyll in that region. So we use this type of a plant leaf for our experiment, right? And we pluck that mosaic leaf, draw a sketch of it with its pattern. So carry out the starch test for it. So after you carry out the starch test, students, what happens? Now you have to get the sketch of it, like using a white paper, get the sketch of that leaf, right? And do the starch test for the leaf, right? What happens? The leaf, because since there are white regions also present, the white regions lack chlorophyll. That is obvious. So therefore, the other regions, the green ones, they have chlorophyll in it, right? So you can understand before the starch test what happened, the white colored areas and the green colored areas are there. But after the starch test, I mean, why mainly why do you involve or do the starch test, carry out the starch test? Because to remove that chlorophyll pigment. After you remove, you carry out the starch test. I mean, after adding iodine solution, the if the color changed, now you can see this is the structure of the mosaic pattern of a leaf, then the color has changed. The white regions are white. There's no color change occurred. It's normally white. Then the other remaining, the green, has changed to a color of kind of a blue, or they may refer it as purple, no? Yeah, purple, dark purple, blue color or purple color. So this color change will indicate that the presence of starch. So this shows that you need chlorophyll for photosynthesis. And even from a mosaic pattern of leaf, you can understand that the chlorophyll is lacking in some regions. They're not there. So the other regions of the leaf, they contain chlorophyll, right? Let's now go over this part a little bit. The white color region lacks chlorophyll, so photosynthesis has not taken place. That is obvious. Therefore, starch has not been produced. So it can be concluded that chlorophyll is essential for photosynthesis. That's why we do this certain experiment, right? Now I'm gonna, all right, okay. Within 2 minutes, let's have a break. Okay, then you all can come back at 7:15 for the theory, right? I mean, we'll continue. There's a certain video I'm ready with a video to show you all. So that the video is about this one of the investigation, because since we are running out of time, we couldn't show you all the videos, right? At least one of the video, especially about, I think so it's about the carbon dioxide one, since students get confused with this carbon dioxide. So I'll show a video on that. Okay. Now we have done how many practicals? Almostly 4. Carbon dioxide, light, and three. Yeah. So the fourth one is about investigation of oxygen. Is oxygen produced? Now it's an illustration or an investigation to show that whether oxygen is produced during photosynthesis or not. Right. Now this part, students, will do it after the break. So we'll get the break around 7 and you all can come back at 7:15. Right. All right. Okay. So we'll start the activity. Right. Okay. Okay, students. Two of y'all are there, no? Where's the other guys? Right. Okay, we'll start then. Now it's the experiment on how oxygen is produced during photosynthesis. Oxygen is a byproduct. So let's investigate how oxygen is produced during photosynthesis. Right. Simply, let's go over the materials used. It's a trough. Trough is kind of like, students, when you can use a kind of a large container or a beaker. It's a type of a large beaker. So trough, a boiling tube, a glass funnel, aquatic plant. So an aquatic plant like Hydrilla, Vallisneria, Salvinia, any type of aquatic plant. Aquatic means kind of plants that grow in uh, water. And you need water. These are the materials required. So this is the setup you have to make for this investigation. You have to get the trough, fill it with water fully, like not fully in sense around like 75%, not up to the brim, not up to this, right? Then you have to place the funnel inverted, upside down, we say. Inverted or upside down, right? Especially they have mentioned the term inverted. So to place it inverted. And then before placing the funnels, make sure students to put the plant, the aquatic plant inside of the funnel. Then you have to place a test tube that is also again inverted. Both the funnel and the test tube, both should be inverted, right? So the purpose of doing this is we have to observe the oxygen bubbles. Let's say if there's photosynthesis in it, therefore oxygen bubbles are released. So we have to investigate on that, right? Let's look at the investigation. So this is a simple practical, right? Definitely when you place the plant to the sunlight, right? So there, or it can be light, any type of light, because we can do this practical even in the lab, right? Using the lamp or a light source, that is also fine, right? After you do it, what happens? After some time, the photosynthesis, now the water is provided, and then yeah, light energy is provided, so essential factors are provided. So therefore they will release oxygen. So these gas bubbles, you can see some bubbles which is released from the funnel. So that indicates, that shows that that is oxygen, because during during photosynthesis the only gas released as a product or a byproduct, it is oxygen. Okay, right. Let's look at it further. Right. As I mentioned, gas bubbles released from the aquatic plants, and that gas gets collected at the top of the boiling tube. You can look at here, top of the boiling tube. Right. Now, how do we measure? Actually, students, they use a test tube. From the test tube, you can't measure the exact volume of oxygen. Right. So for that, we suggest is you can use a measuring cylinder. Measuring cylinder, you can get in the laboratory. It's kind of like a long cylindrical shape. And there are some calibration scales like this, measurements. Right. So therefore, you have to keep it inverted, similar to the test tube. But the thing is, you can measure the volume of oxygen. Remember, don't make it confused, saying that this is the volume of oxygen. That is wrong. The air occupied, the space available, this is the volume of oxygen. Okay, right. Right. Now to indicate is it exactly oxygen or not, the identification test. I think these identification test, students, you all have done in grade 10. So simply what you do, you take a burning splint or a splinter, then you put it inside this test tube with oxygen. Then if it relights, that shows that relights kind of means like kind of sparking, right? Then that shows that the oxygen is present. That is the confirmatory test. Now, is that clear for y'all? The experiment to show oxygen during photosynthesis, production of oxygen. Oxygen is produced during photosynthesis. Is that clear? Y'all can quickly send chat messages, right? One person have raised his hands, right? Good. Yeah, it is clear. Right. Okay. And perfect. Right. So what were the 4 practicals we studied? Number 1, it's about the presence of carbon dioxide, the presence of light. We need light for photosynthesis. Okay, great. Yeah. Then the second one, we studied about presence of carbon dioxide. They need carbon dioxide. So the main thing what we do, add KOH, potassium hydroxide. So in this case, and then the number 3, that's the using of the mosaic patterns of leaves, right? And the very first one we studied about the starch test. Do we carry the starch test for the last practical? No, not necessary, because that is just to show that oxygen is released during the photosynthesis, right? Hope that's clear. Now, students, this is actually the whole topic is over. This is a very simple topic. It's about learning these 4 practicals and the idea of the practical, especially what's the conclusion, what's the observation. Mainly when you do a practical, you will have to know that the main observation, what happens, it can be a color change, it can be a release of gas bubbles or anything, any observations, or and the final conclusion. So from that particular observation, what type of conclusion you can get, right? Now let's study about the importance of photosynthesis. Why this photosynthesis is important? How does it helps to the whole biodiversity, to organisms, right? Let's look. Number 1, light energy is converted to chemical energy during photosynthesis. Plants produce food, and all organisms live on Earth depend directly or indirectly on that food. Yes, so what is the modes of nutrition we studied earlier? The autotrophic and heterotrophic. So in case of the plants, they are completely autotrophic. They don't depend on any other organisms. They make their own food. Okay, right. Therefore, this process is carried out by green plants. So without plants, animals or we humans cannot survive. Why? Because without food, we can't survive. So the main source of food is done by plants. Right. Second, the oxygen gas is required for the survival of aerobic organisms. So especially the organisms that need oxygen to breathe, for respiration, that is aerobic organisms. And combustion of materials it's released mainly by photosynthesis. Combustion of materials means, for an example, to burn something, a hydrocarbon, it can be firewood even. You need oxygen. Otherwise, without oxygen, actually combustion or even for the survival, it's very difficult, right? So who releases this oxygen? Actually, this oxygen is released by the plants. Now, it's like these, students. Now, we breathe in oxygen, release carbon dioxide. We breathe in oxygen and release carbon dioxide. So when we breathe in oxygen, we get that oxygen from the plants during photosynthesis. So whatever the carbon dioxide we release during respiration, that carbon dioxide is absorbed by the plants for their photosynthesis. So therefore, the photosynthesis, you can look at here, carbon dioxide that is accumulated due to respiration and combustion is removed from the environment by photosynthesis. So the final conclusion you can come that where the plants, they are very, very important in order to reduce the pollution. The pollution can be reduced by plants. Why? Because if there is pollution around your surrounding or environment, where if you try to plant more plants and trees, it will absorb all the carbon dioxides. So that will be a carbon dioxide free environment. Okay, and then they help to maintain the carbon cycle as well. Yes, this carbon cycle is not necessary for all, right? Now, is that clear? Actually, this is the overall idea of topic 2. But you all can realize the first topic, we took around 3 weeks to finish it because there were a lot of theories in it. But here, the theory is very summarized and concise. The main idea is you need to know the activities, the practicals related to it. Right. Okay, now let's look at the summary a bit. Number 1, light energy is converted into chemical energy by green plants during photosynthesis because light, so in the form of light, they convert it into chemical energy. That is actually done by the chlorophyll, which is inside the plant leaves. Number 2, light energy, water, carbon dioxide, and chlorophyll are required for photosynthesis. Number 3, the main product of photosynthesis is glucose, and oxygen is produced as a byproduct. That is very important, students. Not only that, I additionally included that point, which is very important. When the glucose is produced, actually it is stored in the form of starch in plants, and it is transported in the form of, especially the translocation, which happens in the phloem, which is known as sucrose. So glucose, starch is the stored form, and then the sucrose is the transportation form, or the how it's transported throughout the plants and the plant stems or whatever. Yeah. So this is the equation. The other thing you need to remember, carbon dioxide. Know to write the balanced equation. So it's very simple. 6 carbon dioxide and 6 water will make up only 1 molecule of oxygen, and it releases 6 molecules of, sorry, 1 molecule of glucose and 6 molecules of oxygen. It makes only 1 molecule of glucose, right? It's like you can use the rule of law of conservation of mass to balance the reactants and the products, right? And the last part, they clearly say that the global importance of photosynthesis is provision of food to all organisms. It can be directly or indirectly, and also maintaining the oxygen and the carbon dioxide balance in the atmosphere. That is the other part. That's why the oxygen is released by the plants and then absorb the carbon dioxide, what we respire, what we breathe. Right. Now that's over. Topic 2 is over. Let's yeah. So there are some questions as well. Then topic 3, mixtures. Then we will go ahead with the topic 3 next week. Right. Now is the importance, the summary. If there's any doubts, you all can let me know. I'll try to clarify that point. Even it can be the whole topic 2, any sections of topic 2. This is an important topic. It can be at a point in investigation. If you all want, you can unmute your microphones and speak. If you are not hesitated to do that. Whole lesson is clear, right? That's great to hear actually. Right. Yeah. What about the other students? Chenumi, thank you for your reply. It's clear. Okay, right. Thank you for your reply as well. What about Amar? Sorry, I just say, I think I'll call him as Asmat. It's clear, right? Okay. Thank you very much for your reply. Right. Okay, great. Now, students, what we have to do is, first, let's write the definition of photosynthesis and let's try to write some very short notes on this particular topic. Right. Now, write the topic for today, photosynthesis. You all have to be very quick in writing notes because I'll move so quickly. Right. Then, first, let's write. So since you all are year 11 students, grade 11, Okay, 1 second. One student left. Okay, right. So what you need to know is let's write about the modes of nutrition. Okay, right. Now let's write the first note, students. I think you all are good in writing notes when I read, right? Is hope that's okay. Right. So let's start. There are mainly two modes of nutrition. There are mainly two modes of nutrition. Number one, it is autotrophic. Autotrophic. This is the spelling actually. Autotrophic. Autotrophic mode. Autotrophic and heterotrophic. Okay, so then we'll write modes of nutrition is divided into two types. Autotrophic, heterotrophic. Right. Autotrophic means where they plants. Examples are plants. Where they produce their own food. I can write that quickly. Where they produce their own food. Where they produce their own food. Okay, right. And they do not depend on any other. If there's anything, if you are missed out or if you can't follow up, please let me know through the chat or you can unmute your microphone. So they do not depend on other organisms. They are independent organisms. They do not depend on other organisms. Right. Heterotrophic animals, animals and humans, they belong to the heterotrophic, where they are dependent. They cannot produce their food. Dependent and they cannot produce their food. Right. Then that is done. Now we'll move to the other part. The essential factors. What are the essential factors needed for the photosynthesis? You all can copy these 4 factors quickly. You have no enough time. right? Chlorophyll, light energy, water, carbon dioxide. Okay, hope that's all okay. Now we'll move to, actually students, I don't like to give like the same exact notes what's given in the book. Every time what I do is like use the basic simple techniques in order to make notes and also for your understanding. So the usual students, they know about me. One of the students have not joined also today. But normally what I do is I give very summarized tables, flow charts in order to make it easier, right? Okay. Now, before that, we'll make a quick note on this as well. The glucose produced during photosynthesis will temporarily stored in leaves as starch, then converted into sucrose and transport into the other tissues through phloem. Right. This is a very important part, student. Please note this. Okay, I will normally give the layout of a table. Students know about it. The usual, the ones who normally join the lesson, the layout will be provided for them. So what you have to do, you have to refer to the notes and then you have to fill the layout, right? Quickly finish this, then we can start I'll create the layout and explain you all. Okay, I think y'all are done with it. Now, students, I'll give the layout. Okay, right, great. Right, now it's actually there's just 5 minutes left. Therefore, I will show the layout. You all can draw the table. Normally we do it in the Word document, but since we are running out of time, couldn't do it. Right, now it's like this. We have to divide the table. Hope you all can see the board. I'm going to divide into columns, especially the practicals and the first column. Normally in a practical students, you all have to understand the materials needed. Those things are not that necessary, right? So the thing is mainly chemicals means like importance of some chemicals you might know, like KOH, that is important. So like that. But the thing is, what is the observation you observe and the conclusion? That is the main thing when doing activities or practicals, right? Now you all can do it as a homework. Now in this case, let's see the activity, and then observation. Whatever you observe, it can be evolution of gas bubbles, it can be even color changes, right? And finally, how do you conclude? And a conclusion. This is the overall idea of these investigations, right? Now, what are the activities we did? Number one, the starch test. Okay, starch test. So the observations are like what happens if starch present. Normally in the starch test, the observations are, yeah, that's a conclusion actually. Color changes to black, sorry, it's blue or purple. Blue or purple if starch is present. Color changes, yeah, here that is enough. Then the conclusion indicates the presence of starch, right? Then you can follow up the other practicals as well. The second one, uh, test for um, light, sunlight. What is the main no color change in the black covered leaf, and color change occur, color change in white polythene covered leaf. Okay, then what is the final conclusion we can come up? Okay, final conclusion. Sunlight is necessary for photosynthesis, right? So you all can do the rest like this. I have given the layout. You all can do for the other tests as well. For the chlorophyll, chlorophyll spelling, yeah. Then the other one, it's the production of oxygen, O2. Right, you all can fill it. Okay. Now, actually, we'll conclude the lesson. That's the theory part. So we have to write few notes as well. We'll continue in the next lesson, right? Cherimi, you don't need to worry. I can see that you have a connection issue, right? Uh, that's fine. We'll send the recordings for you, right? Okay. And yes, is there any doubts? Quickly, you all can clarify. Now, then we can move to the paper lesson. Every students can join the paper as well. If you are willing to join, you all can stay because we don't give a break in between. Already a break is given. So we'll Shall we move to the paper? Jeremy, if you want, you can quickly take a picture of this. These things you have to fill. That's given as a homework. As a homework, these ones. Simple. So this is a simple idea. Okay, right. Then let's, today actually I'm ready with a paper, the 2016 paper. It's the very first year of the first new syllabus. So we are going to do a paper on that. Right. I'll share the paper with you all. Right. First, let's target the MCQ questions. Normally, the MCQ, it's very easy to get the marks because it's one per question. Right. Hope you all can see the screen. All right. Please keep muted your microphones unless if you want to speak anything important to the lesson. Right, students. Now, normally in the paper lesson, you all will given a chance to do the paper, and I will explain the theory behind each questions, and we give review points as well related to each questions. How now? Suppose if you get a similar type of a question again in the paper, right? Then you will be able to do it. That is because of that review questions or the review notes. Right. Now I will give you all around how many questions are there. Don't just underline and keep it. Just try to logically think and underline it. Okay. So the first one is like kind of easy questions, and second, okay, that is these are theory. Many theory questions are there. Hopefully you all can do the first. These are the questions you all can do. Normally some questions are there which is related to grade 11. Still we have not even started. So this first one you all can do. And don't worry, these papers will be sent to you all. Number 2. And then, yes, you all can do this as well. This is a basic thing, yes. Acids and bases. No, you all have not yet studied. So therefore, you can't do this. Coagulation of rubber, this one also you can't do. Amphoteric oxide you can. Disinfectant, no, you all can't do it. This one you can do. 10th one also you can. 11th you can. 12th you can't. Non-granulocytes. Eosinophils, basophils, number of lone pairs. This one you can do. The temperature, this part you can't do, I guess. Isoprene, this is organic chemistry, you can't do. Sexual reproduction, this one you can do. Lenses we didn't still study. 20th we can do. Because even last week we did on that. And this one you can do as well. Amali search. This is about the vitamin deficiency. You all can do it. Photosynthesis, great. 23rd question, it's about photosynthesis. You all can do it. A diagram of longitudinal section of a bisexual flower. Bisexual flower. That's the part of this one, yeah. You can do it. Neutralization reaction. Yeah. Okay. Then I have ticked some questions which you can do, and the questions which you can't do. So those questions please ignore and try the questions which you can do. Now in this set, the first set, you can see here, um, the first 10 questions. Out of that, please ignore the eighth and the fifth. Fifth and eighth should be ignored. Unless all the questions, please try. Everyone. And send your answers through the chat. Let's see who got the highest. No, no. The questions that you don't need to copy in the book, Putap. You can just write the answers. Just it's MCQs, no. So you can just select the correct answer. For example, students, yes, that's good. You all asked. You all can just write question number one, then the answer. You can just select the correct answer. Not necessary to write the questions because no point. And I'll be sending these papers to you all. So please ignore the question 5 and 8. Right. All right. After you all have done, normally 1.5 minutes, then 8 questions are there, maybe like 10 minutes or 15 minutes maximum given for this finished. Oh, wow. Okay. Let's see. So please send your answers through the chat. You all can send, if you all done in the book, you all can send it a picture of what you have done, or you can send just typing it question one, the answer, question two, and what's your answer. And the ones who have done just with guessing, you all will be caught because I'll ask the way you got the answer. If it's theory wise, that's fine. What about other students? If you all have finished, please let me know. Then I don't want to wait again. One student have finished. Give me an update through the chat message saying that you're finished. Okay. One went. Almost. All right. Your answers you have sent. No. 20. I didn't give you 20 questions. No. Can you see 20 questions in this? I said you to do the first 10 questions. How did you do the other remaining 10 as well? You have the 2016 paper. I didn't say which year paper as well. Yeah, I said, right? Yeah. You don't have the paper. Then how did you do the other remaining questions? Because I'm screen sharing only the first 10 questions. Ah, you studied at school. Ah, right. Okay, what about the other students? I'll give another 3 minutes for y'all. Chenimi is left, no? So Shima, have you completed? I'll give 2 minutes. Done, Shima? Is that okay for as a reply for you have completed, or do you need extra time? You are completed, right? Okay, then let's start the discussion. Question one, right? Shima, you can feel free to send the answers to us just to check. Previously, as one student has sent. If it's possible, yes, you can send it to the chat. Okay. Or you can send it through the WhatsApp number which they have sent you. You can send it through that, a picture of whatever you are done. Right. Now the first one, students, which of the following is a vector quantity? Right. Now when it's a theory of vector quantity, you will have to remember that there are two quantities in physics. Quantities can be two types, vector and scalar. Right. Therefore, what is known as a vector, students? Vector means the quantity that contains both the magnitude and a direction. What is known as a magnitude? Now, for an example, I take two quantities, especially one is velocity, the other one speed, but they are from different quantities. Velocity is a vector. Why do we say velocity is a vector? Right, good. Thank you, Shima. Right. So why do we say velocity is a vector? Because it has a magnitude. What is a magnitude? It is a value, an exact measurement they give. That is known as magnitude, and a direction as well. Normally we say an object moves to a direction to the left. It can be to the east, west. So that is the reason velocity belongs to the vector quantity. Then the speed. When it comes to the speed, students, we say the car traveled at 60 kilometer per hour. The vehicle traveled at 100 kilometer per hour. But we don't specify a direction to it. That is the reason the velocity belongs to the vector quantity and the scalar quantities are under the where only the magnitude is there. So only magnitude present, that is a scalar quantity, and if there is both magnitude and direction, that is a vector quantity. Is that clear, students? Okay, so let's look at what's the answer, the best appropriate answer. Which of the following is a vector quantity? So the answer can't be distance, it can't be speed, it can't be time as well, because they have only magnitudes in it. So the answer should be weight. The answer should be weight. Let's see how our students have answered the question. Asmat, the one. Good. What about Shima has written the one first answer. Good. Next one. According to the standards of binomial nomenclature, that is a naming of organisms. We name organisms. Select the option which gives correctly the scientific name of jungle fowl. Right. Normally, there's a set of scientific names. Gallus lafayette. It's the name actually. But how do you write it in a standard way? These are the rules you have to follow when writing in the standard way. Right? You can't write every letters as uppercase letters. This is wrong. Right. Then the other thing, it is now since it's printed, you have to give it in italicized form. Italicized. So out of these ones, everything, the whole thing should be italicized. So here in this case, one, this is not at italicized. So that is wrong. So there can be two options, either third or the fourth. But how can it be the third, or is it the fourth? Now we have to be very specific. The third should be the answer because we write the first, the genus and the species. So this is the genus. the genus should be written in a capital letter. The first letter should be capital. Assume this. This one is capital. That is very important. Okay. And the second letter, it is not necessary to write it in capital. Therefore, the best answer is the third answer. Did you all understand, students, about the scientific writing, the binomial nomenclature? Is that clear now when I look at the answers? The third one. Good. Both students have done well. Right. If it's clear, I think it's clear for 2 students. What about Chenumi? Oh, she's still joining. Right. Next one. The gas which is used to produce margarine from vegetable oils is. So the answer should be, student, that's a direct answer, a theory based. It's hydrogen. Normally, you can study about the uses of gases. In grade 10 chemistry section, you have 3 types of gases you have to study. Hydrogen, carbon dioxide, and the other one, hydrogen, carbon, oxygen. Those are the 3 main gases. Actually, there are uses, chemical and physical properties. So therefore, you have to study on that as well. Okay, I will just show a small layout of what we have done with the student. 1 second. That is easy for you all to study about the gases. One second. Right. I will share one of the work we have done with the students. It was a grade 10 student actually. It's just a kind of a layout. Right. You can look at here. This is the layout I mentioned. When you compare the main gases in the atmosphere, you can compare kind of tables like this. It can be useful for any student. So, especially in your syllabus, they have targeted the physical and chemical properties. Actually, I have not listed it, it's just the layout. Then you can list out the oxygen, carbon dioxide, the physical and chemical properties. They vary from one gas to another. Zoom the screen. Okay, but I'll do it. Right. Hope now, oh, it's too much. Hope this is a bit bigger than before. Okay. Then let's look at this one. As I mentioned, you can list out the physical and chemical properties, right? And the composition. What is composition? How much of these gases are present in the atmosphere? Normally, oxygen is 20%. Carbon dioxide is 0.03%. That's a very small percentage. But again, hydrogen, it's much smaller than carbon dioxide. Right. And then students, there are actually, these are grade 10 sections. I'm trying to recap y'all. There are methods of collecting these gases. Upward displacement of water, downward displacement method. You all can classify that also into your summary table, the methods. And there are certain reactions as well. What are the different chemical reactions you can use in order to produce? Normally, for the production of oxygen gas, there are many steps. Now, the main one is they use potassium permanganate. They heat it strongly. So therefore, they get K2MnO4, MnO2, and oxygen is released. When a gas is released, we indicate an arrow upwards. Right. This is strongly heated. So this is known as thermal decomposition. Thermal decomposition, or we can chemical decomposition also correct. The reason because we strongly heat it. By the influence of heat only, the reaction happens. Right. And there are some certain other types of reactions which happens to produce carbon dioxide, hydrogen. You all can list that. Actually, my focus is not to do that, just to show that when you want to study these types of gases, you can use these type of identification test in one uh, column, and then method users. Then when it comes to users, students, you all can type many users and compare them, right? Is that clear, the summary table of gases? You can send quick chat messages. Asmat and Shima both. If it's not clear, please let me know. Hope that's clear. Then I'll move to the paper. Clear. Good. Shima, it's clear. So Asmat, is that clear? He's not there, maybe. Okay, I don't know. Right. So that is the third one, right? Which of the following seeds is adapted for dispersal by wind? Normally dispersal by wind, apple, rubber, bitter gourd, bar. The answer should be, what are the answers you all have given? The third, it's a fourth, no? Fourth one. Bar, yeah. So the ones which are dispersal by wind is students like this. Bar means, I think the name, the common name is given. Wait, let's look. Yeah, kind of like they have feathery things. So the answer should be var. Okay, right. Kind of like feathers around it, so which helps it for the dispersion. Dispersion means moving of these seeds from one place to another. Big the influence of wind, because it's dispersal by wind. This one you can't do. And yeah, third and fifth, not the third, fifth and sixth you can't do. Now seventh, which of the following is an amphoteric oxide? The answer should be aluminium oxide. That is in grade 10, first topic. Not the first topic, third topic, I guess, chemistry. Amphoteric oxides are aluminium oxide. These are acidic oxides. These everything are acidic oxides. You all can note it if you want it. Acidic oxides. Chlorine, Cl2O7. Acidic. This is the one which is amphoteric. And then sodium oxide is not acidic, it is basic oxide. Normally metals, they form basic oxides. Non-metals, they form acidic oxides. But aluminium, even though it's a metal, it is forming an amphoteric oxide. Shall we write a note, students, on that? Shall we write a review note on that? Right. So let's write. Oh. Right. Let's write it. Okay. Like, let's see whether Chinumi can join. Right. Okay. Chinumi, can you hear us? Hope you can hear. Right. Now, we are going to give a note on this amphoteric oxides. I mean, oxides, especially. We'll write a quick note on that. Right. So that's for the question 7. Right. So simply write all the metals, most of the metals, sometimes if you have connection issues, you can't hear me. So therefore, I'll type it again here. Most of the metals they form basic oxides and non-metals, they form acidic oxides and only aluminium forms amphoteric oxide. Amphoteric oxide. Therefore, okay, let's write a note on amphoteric as well. Okay. The properties in between metals and is known as amphoteric. That's like in between the metals. They have the both, not in between. Yeah, in between, kind of in between, or both, a mixture of metals and non-metals is known as amphoteric. Right. All of y'all can copy this note. That's for the question 7. For the question 7. Right. Right. Why didn't you inform me earlier? So you shouldn't, I mean, you could have listened to the recording from the first itself. We normally record, even Sugitana didn't join and she asked for the recording. But the thing is like this, if you have asked for the recording, then anyway you will marked as a person who is attended the lesson. Suppose if you not need the recording, then we'll give you an extra class. Those are the two options they offer in the academy. So yes, we are almost recording it. Yes, we can send it. Shima, if you want to leave, yes, definitely. You can leave. Yeah. Okay, right. Goodbye. Right. Hope you all have copied the note. Mm. Now let's do the other part of the paper. Right. The other question. Actually, students, you all can't do the 8th one. Still, we have not studied about disinfectants. The number 9. The catalyst which is used in the manufacture of sulfuric acid by the contact method. That is, students, when you learn in the what's that topic number 17 of grade 10, you learn that there are 4 factors: temperature, pressure, concentration, and then the 4th is catalyst. They increase the rate of reaction. When you want to increase the chemical, the rate of reaction, actually you have to increase these factors: surface area, and the other one, it's a surface area. So I'm going to give you a small recap. Temperature, actually when the temperature increases, the particles kinetic movement is higher, so therefore they move faster, they collide with each other and increase the rate of reaction. Remember one thing: for the rate of reaction to increase, the main thing, they should collide together. That when there's more collisions, the rate of reaction increases gradually. Right. The number 2, concentration. Again, when there's more concentration, more particles, so therefore they collide and it increases the rate of reaction. Number 3, pressure. Pressure is only exerted by gases. Pressure is only exerted by gases. So therefore, pressure, when there's more gas molecules, they try to collide with each other and increase the rate of reaction. And remember, in a small volume, if you try to fill the gas, there is greater pressure. Why? Because there's more collisions within a smaller volume. If the volume increases, so the pressure will reduce. If the pressure reduces, the rate of reaction also reduces. Okay, right. I'm going very quick, I know, but I can't help. These are the things from grade 10. Just at least if I give you the topics, you all can revise it. Right. And then the fourth one, surface area. So for an example, if you take a sample of powder of a metal or anything, any chemical substance, and if you take a lump or a strips of it, then that which will have a smaller surface area when it comes to the lump or a strips. But if it's a powder, it has more particles. The size is smaller. Therefore, the surface area is greater. The rate of reaction is greater. These are the main four factors. Additionally, there's another one which is known as catalyst. All the students, please study on catalyst, which is a very important part. So in that particular topic, there is a theory where each of the use of catalysts, there's a certain table. So in that, you need to understand to manufacture sulfuric acid, they use vanadium pentoxide. Vanadium pentoxide. Okay, so the answer should be vanadium. So what does the catalyst do? It increases the rate of reaction by producing an alternative pathway. By using an alternative pathway, it increases the rate of reaction. Right. So when you want to increase the rate of reaction, if you put a catalyst to the reaction, it will increase the rate of reaction. That is the purpose of increasing the using a catalyst. Okay. Understood? Yes, you understood. Right. Okay. Right. Then we'll move to the other part. It's actually theoretical parts to the monocotyledonous plants. Okay. Monocotyledonous plants. So in this case, you all have to understand there are two types of plants actually. Dicots and monocots. So what you all have to do, find out the features of the monocots and dicots. Monocots and dicots. Now, if I try to find out, it will take time. So trimerous, pentamerous, parallel venation, reticular venation. There's a lot of things. So based on that, monocots, dicots. Monocots mean single cotyledon, single, but here there are two cotyledon. Dicotyledon means two cotyledon. One single cotyledon is present, cotyledon. Okay, single cotyledon, and two cotyledons are present in the dicots. Right. Then trimerous. Trimerous means, students, like this. Three petals are there in one flower. Three petals are attached. Trimerous. But this one, dicots, are tetramerous. Four petals are there. And pentamerous. Okay, tetramerous or pentamerous. And parallel venation monocots, parallel venation, they're parallelly, their veins are located parallel venation, and reticulate venation in dicots. These are the different examples you need to know. All right. So then the answer should be, I'll share the screen anyway. Bare trimerous flowers possess leaves with a no, reticulate venation can't be. Taproot no, so secondary growth. So answer should be 1. Bare trimerous flowers. Monocotyledonous, they bear trimerous flowers. Okay, right. That should be the answer. So where did it go? All right, it's still showing. Right. Then the number 11. In which of the following instances does a chemical change occur? What is known as a chemical change and a physical change? Simply physical means due to a like boiling, melting, freezing. These are the physical changes that happen. Chemical changes means chemically it should happen. Now, for an example, in vaporization of water, what is vaporization? Vaporization means they boil water, right? So, for example, you heat the water and it starts to boil. Vaporization converts into a vapor. Water liquid is converted into a vapor, a gas. In burning of firewood, right? In burning of firewood, right? In melting of mothballs, in melting of wax. Actually, the answer should be burning of firewood. It is the one, the chemical change, because it is known as combustion. It is known as combustion, right? So it is known as combustion. Hope that's clear. Right. Then you can't do the 12th because still we have not studied about the ecology part. Right. Okay. Then you all can try 14th. Yes, Chenvi, you can try it. That's good. 14th. What about 15th? 16 degrees and what is the temperature difference? Kelvin and still we didn't study. Yeah. So 14th you can do. These are the questions you can do. 17, asbestosis, bronchitis, gastritis. This one I think we have not yet studied. Yeah, you can try the ones which I have ticked. Yes. Yeah, Chenumi, actually I'm not available today. There's another lesson after this. So what's the best time I can give you to contact is maybe tomorrow, even tomorrow it's a Friday, right? Yeah, tomorrow you just drop me a message. If I'm free, I'll definitely call you back. Right. Yeah, no problem. Give me an update after you're done with the questions, then we can discuss. Normally the lesson ends at 8:45. No, dear. If you want, that's only all the students have left. Actually, that's because of their third term exam. If you want, you can leave. Then I have to just speak alone and finish the lesson. Yeah, if you have any other classes or anything, you can leave. Yeah. Anyway, the lesson will just go for about 5 or 10 minutes. So you want to leave the lesson? What? Yeah, if you want, you can leave. That's fine, dear. Because anyway, you're a trial student, no. So the other students, we will normally record the lesson and send it. And if you can wait. Ah, right. Okay, that is fine. Yeah. Right. Then I think it would be boring if it's like too silent. We'll quickly discuss these questions. Number 13, actually, sometime you cannot do, I guess, because still we didn't study about the black part, no? Right. The 14th one. Number of lone pair electrons around oxygen atom in a water molecule. How much is it? Normally, water molecule, this is how the water molecule looks like. This is how the water molecule looks like. So oxygen, hydrogen, 1, 2, 3, 4, 5, 6. Because the idea is oxygen is a group 6 element, right? Group 6 element. So therefore, 1, 2, 3, 4, 5, 6. So therefore, we write a last lesson in grade 11. You mean now or in a extra lesson? The ecology part. If you want, yes, we can do an individual lesson, but now it's not possible. No, dear, it's actually the time you will surely will finish the lesson about within 5 minutes. That's and also it's not practical to finish that. It's that what's the last lesson? It's that biology part, right? Yeah, the biology one. Yeah. So actually, the time is not enough. If you could have informed earlier, the biosphere. Yeah, you can do something. But what you can do is, if you're free, you can let us know. Um, we can do an individual lesson. I'll discuss that tomorrow with you. Okay. Yeah. Because normally this lesson is getting recorded, so better we'll discuss it. Your Friday it's free. Ah, right. Okay, fine. We'll discuss that tomorrow. The thing is, Friday we don't do lessons, but just to discuss how the lessons can be, and let's see how its availability. Maybe like a Wednesday is okay. Let's see. Right. Then we'll go with the other one. So the answer should be the number of lone pair electrons. Okay, no problem. Lone pair electrons. I show the screen around oxygen atom in a water molecule. So in this case, lone pair electrons. So therefore, they say lone pair, right? Lone pair electrons. So yeah, so then that should be 2. The answer should be 2. So there are 4 electrons, but they are lone pair. So therefore, the answer should be 2 lone pair electrons are there. Right. Then the next one, on a certain day, you can't do that. Yeah. Next, 18th one. In the asexual reproduction, a large number of offsprings is produced in a short period of time. Meiosis occurs, gametes are produced, new species with better adaptation to environment are produced. It is very direct. The difference between asexual and sexual reproduction, it is very easy because you have to understand that sexual reproduction it involves in the meiosis, so therefore meiosis can't be the answer. Gametes are produced, right? Gametes are produced. So therefore, that is also in the sexual reproduction part. So that can't be the answer again. And then variation and adaptation is actually in the fourth, that is also in sexual reproduction. So large number of offspring produced in a short time period. That should be the answer. Okay. Right. So the answer should be the first one. Right. I think then we can conclude. I will do this one and conclude it. Okay. Right. Question 20. A diagram of a muscle tissue is given below. Select the option which gives correctly the name of the muscle tissue and its function. So basically, there are three types of muscle tissues. What are the three types? Skeletal muscles, cardiac muscles, and smooth muscles. So you have to compare the muscle types. That's good. Then you'll understand. So they have given a voluntarily, involuntarily. Voluntary means they can function in their own. Involuntary means they cannot function in their own. So in this case, you have to understand voluntarily cardiac muscles are not controlled. They are involuntary. We can't control it. Skeletal muscles involuntary. No, it is voluntary. That's why you can move your arms, move your body, right? Skeletal muscles voluntary control. That is correct. Skeletal muscles voluntary control. Correct. Cardiac muscles involuntary controlled. Ah, they have given the muscle type as well. What is this muscle? That's the thing. It is a, there are intercalated discs. So the answer should be fourth. The answer should be fourth. Right. Then we'll conclude here. Is there any doubts, Shenumi? Is there any doubts? So the answer should be the fourth because this is a picture of a in cardiac muscles and they are involuntarily controlled. All right. Shall we conclude? Right. Okay. Then have a great day. Goodbye.